Good morning, students. Today I am going to discuss session 6 of chapter 5 from your class 12 biology syllabus. The name of the chapter is Principles of Inheritance and Variation. Already we have completed 5 sessions from chapter 5, and from these 5 sessions we actually got to know a lots of things, that the basic terminology of genetics, the basic idea about genetics, Mendel's monohybrid and dihybrid cross, incomplete dominance, codominance, ABO blood grouping system, linkage and recombination, and the pedigree analysis and mutation also. So from this session actually we'll get to know different kinds of the genetic disorder. So let's start the today's class. Actually today we are going to discuss genetic disorder. So first you should know what is the basic definition of genetic disorder. So these are disorders or illness actually which is caused by one or more abnormalities in the autosome or sex chromosome. So I am repeating again. So in our body total 46 chromosomes are there, and within 46 chromosome 44 is autosome and 2 is sex chromosome. So these are actually different kinds of the disorder or illness which is actually caused by one or more abnormalities in the autosome or sex chromosome of the person. Okay, so this is also referred as the autosomal disorder or the sex linked disorder. So this genetic disorder actually divided into two types. The first one is the Mendelian disorder. Okay, I am highlighting this thing. First one is the Mendelian disorder and second one is the chromosomal disorder. So there are lots of diseases are there which is included into the Mendelian disorder. The first one is the hemophilia, second one sickle cell anemia, third one phenylketonuria, fourth one cystic fibrosis, fifth one color blindness, and the last one is the thalassemia. And there are different kinds of the chromosomal disorder is also there, the Down syndrome, Turner syndrome, and the Klinefelter syndrome. So you have to study each and every syndrome, each and every disease, okay, because all those are included in your class 12 biology syllabus. So actually first next thing you should know what is the basic things of the Mendelian disorder, what is the basic definition of the Mendelian disorder. So actually these kinds of disorder are mainly determined by the alternation or the mutation in a single gene. So I am repeating again. So these Mendelian disorders are mainly determined by the alteration or mutation in the single gene. Okay, so already we have discussed the genetic disorder grouped into two categories, the Mendelian disorder and the chromosomal disorder. Clear? So what is the basic definition of Mendelian disorder? So I am repeating again. So these kinds of disorder are actually determined by the alteration or mutation in the single gene. So it actually obeys the principles of the Mendelian inheritance, okay, during the transmission from one generation to the another. So these kinds of the disease are transmitted to the next generation according to the principles of the inheritance from the Mendel's law. Okay, already we have discussed Mendel's 3 laws, first law, second law, and the third law. Okay, so this is actually the main thing of the Mendelian disorder. So Mendelian disorder traced in the family by the pedigree analysis. Example: hemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketonuria, and the thalassemia. Okay, so already in our last class we have discussed pedigree analysis, and through the pedigree analysis actually we get to know the inheritance pattern, different kinds of the genetic disorder. So this Mendelian disorder is also determined by the pedigree analysis. Okay, so first you should know that these kinds of disease are either dominant or recessive. So how you will identify that this is dominant or recessive? Through the pedigree analysis. Okay, so actually there are 4 types of the disease. This can be dominant or recessive. The first one is the autosomal dominant. Okay, second one is the autosomal recessive, third one is the sex linked dominant, and the last one is the sex linked recessive. So trait may link to sex chromosome, that's called the sex linked dominant or sex linked recessive. And X linked recessive trait, it means the transmitted from carrier female to the male progeny. Because why this is carrier female? Why this is female? Because the X chromosome is dominant to the female body. So this is the basic things of the Mendelian disorder. So I am repeating again, what is the basic definition of Mendelian disorder? So these kinds of the disease actually determined by alteration or mutation in a single gene. And how you will identify this thing? By the pedigree analysis. There are several types of disease are there which is included into the Mendelian disorder, like the hemophilia, sickle cell anemia, color blindness, all those things. Okay, and all those things are included in your syllabus. So first disease is the hemophilia. So I think all of you are quite familiar to this disease. So it's actually the sex-linked recessive disease. This is actually the sex-linked recessive disease. What is the meaning of this? That this is actually transmitted from an unaffected carrier female to some of the male offsprings. So what is the meaning of this? Now you can ask me one thing, man, what is the meaning of the carrier female? Okay, what is the, how you will define the carrier female, and why female is only become carrier? Because female body has total 2 types of sex chromosome, X and X. Okay, so and the male body has 2 types of sex chromosome, X and the Y. Okay, X and Y. So when, so female always will be carrier. Why? Because if the one X chromosome become diseased, then another X chromosome can express. So that's why this X chromosome can mask up the others X chromosome's character. So this is called the carrier. Okay, that's why this is, because this actually the position of this X chromosome in the homozygous in condition. But when the male body, suppose one X chromosome, suppose this X chromosome the diseased, but Y chromosome cannot mask up the character of the X chromosome, or suppose Y chromosome become diseased, so X chromosome cannot mask up the other characteristics. Okay, so that's why male cannot be carrier. Okay, so the defective individual continuously bleed to a simple cut. So due to this disease, actually a patient continues bleeding even to a minor injury because of the defective blood coagulation system. Okay, so that's why this is also called the bleeder's disease. So what happened in this disease, what happened in this case, that due to this patient, actually due to this disease, the patient actually continues bleeding even to a minor injury. Okay, because of the defective blood coagulation system. So the gene for hemophilia is located on the X chromosome. I am repeating again, the gene for hemophilia is located on the X chromosome. Okay, so in this disease, a single protein that is a part of the cascade of protein that involved in the clotting of the blood is affected. So these kinds of disease transmitted from unaffected carrier female to some of the male progeny. So this is the basic characteristics of the sex-linked recessive disease, that these kinds of disease always transmitted to the carrier female to the male progeny, not the female progeny. Okay, so this is the heterozygous female transmit to the sons. Okay, so the female being hemophilic is rare, mother should be carrier and the father hemophilic. So when female will be hemophilic, when the mother is carrier but the father is hemophilic, that time female can be hemophilic in nature. Okay, so this is the basic thing. The female suffers from this disease only in the homozygous condition. Okay, the family pedigree of the Queen Victoria has this disease, that's why this is also called the royal disease. That's why this is also called the royal disease. So this is the basic things of the hemophilia. So what is the meaning of hemophilia? This is one of the disease by which the blood coagulation system become disrupted. Blood coagulation system become disrupted. Okay, so in this disease, a single protein which is a part of the cascade of the protein involved in the blood coagulation system, blood clotting system, is affected. Okay, so this is the basic things of the hemophilia. This is one of the most important sex-linked recessive disease. So this is very important for your exam. Next thing, actually I have made one diagram, that this is the father with the hemophilia. Okay, so this H means the hemophilia and the mother is unaffected. Okay, so the possible outcomes is when you are crossed to father and mother, it means the XXH. Okay, so this is all daughters will be carrier, not the hemophilic, and the next one is the normal son, X and Y. Okay, so this is the basic things of the hemophilia. And another thing is the father is unaffected but the mother is carrier. Okay, father is unaffected but mother is carrier. So I have drawn two possibilities. Okay, so first possibility, this is the first possibility. The first possibility is the father is hemophilic but mother is normal, mother is unaffected. And this is the second possibility, that the father is normal but mother is not hemophilic but carrier. Okay, so what happened? First XXH. Okay, so this is XXH, so it will be the carrier, then XX, then the normal daughter, then the XHY, this is the son with the hemophilia. Okay, because there is no actually the gene who can dominate the X gene. Okay, and next is the XY, so this is the 20% unaffected child. So there are first of all the 25% unaffected child, 25% daughter is not carrier, it means the totally unaffected, 25% son with the hemophilia, and the 25% daughter will be the carrier. Okay, so this is the basic pedigree analysis. Through the pedigree analysis you will get to know in this way. Okay, this is the third possibility. This is the another possibility, that the father with hemophilia and the mother is carrier. Okay, so the first one is the XHXH, it means the daughter with the hemophilia. Okay, then XHX, so this is the carrier. Okay, next is the XHY, it means the father with the hemophilia, and next is the XY, so this is the son without hemophilia. So when we cross the hemophilic father with the carrier mother, so then we will get these four types of offspring. That 25% offspring will be without hemophilic. Okay, 25% offspring will be this daughter will be hemophilic, and 25% son will be the hemophilic, and 25% daughter will be the carrier. So this is the third possibility. So in this type question may come during the time of your exam. Next thing is the sickle cell anemia. Next disease is the sickle cell anemia, and it's actually the autosomal linked recessive disease. So this is not the sex linked, this is the autosomal linked recessive trait. Okay, which can be transmitted from the parents to the offspring when both the parents are carrier for the gene. Okay, so this is the basic thing. So this is actually the insertion and the deletion of some base pair. So for that reason the sickle cell anemia will occur. So this disease actually controlled by a single pair of allele HBA. Okay, so gene is HB, hemoglobin gene. Okay, so only the homozygous individual, which is HBA, show the diseased phenotype. Okay, so I am showing this thing. This is the autosomal linked recessive trait. Okay, and it's controlled by the pair of allele HBA and HBS. So homozygous individuals of HBS it means the diseased, and heterozygous individual HBA HBS, it means the unaffected but the carrier. When it's actually positioned in the homozygous condition, it will be diseased, but when its position is the heterozygous, then it's become unaffected. So defect is due to the substitution of the glutamic acid by valine at the 6th position beta globin chain of the hemoglobin. So this is the basic thing. Okay, so I am showing the picture. So the first thing is the glutamic acid. Okay, this is the glutamic acid. Okay, but the change is there, that CAG, first one is the CTC, then it's CAG and the GTG. Okay, so this will be the valine. Okay, so this is the two substitution actually. So it's actually affected, this defect is due to the substitution of the glutamic acid by the valine at the 6th position. Okay, just 1, 2, 3, 4, 5, 6. Okay, so this is the basic things of the sickle cell anemia. This is the basic things of the sickle cell anemia. I am repeating again that the 1, 2, 3, 4, 5, 6. Okay, so this is the 6th position and this is also the 6th position. Clear? So in the 6th position, the first item was glutamic acid, but during the time of the changes, it's become the valine. Okay, so this is the basic things of the sickle cell anemia. So mutant hemoglobin, the polymerization under low oxygen tension causing changes in the shape of the RBC to the sickle cell like structure. So actually the symptom is the highly anemic patient. Through the sickle cell anemia, actually it's it will be the highly anemic patient. Okay, and this gene, this HB gene actually located in the 11th number of chromosome. Okay, so this defect actually beneficial to people which living in the malaria prone areas. So this is the sickle cell anemia. First you should know one thing, that this is the autosomal linked recessive trait. Okay, don't mix up that the hemophilia is the sex linked recessive trait disease and the sickle cell anemia is the autosomal linked recessive trait. So this is the actually the picture. Okay, so this is the father with trait and the mother is normal. So this happened here, father with trait and the mother with trait also. So this is the basic thing. Okay, next thing is the PKU. Next thing is the phenylketonuria. Okay, next disease is the phenylketonuria. So actually it's an inborn error of metabolism. So short form of the phenylketonuria is PKU. Okay, so it's an inborn error of the metabolism which is actually inherited as an autosomal recessive trait. So this is an autosomal recessive trait. So this PKU is one kind of the autosomal recessive trait. So this is one inborn error of the metabolism, and actually it affects the individual which lack enzyme that convert the amino acid phenylalanine to the tyrosine. So this gene actually located in the 12th number of chromosome. So it's actually the rare disease, but how is this disease actually takes place in our body? So it's actually rare disease in which the individuals lacks an enzyme called the phenylalanine hydroxylase. I am repeating again, phenylalanine hydroxylase. Okay, so the lack of this enzyme which is needed to the breakdown of the amino acid, and it transformed to the phenylalanine to the tyrosine. So this phenylalanine actually accumulates and convert to the phenylpyruvic acid and the other derivatives. Okay, so the accumulation in the brain result and it forms the mental retardation. So this is the symptom. Okay, so this actually is needed, the breakdown of this amino acid phenylalanine into tyrosine in the liver area. Okay, so the phenylalanine is accumulated and gets converted into the phenylpyruvic acid. Okay, so these derivatives leading to the mental retardation during the accumulation of this thing in brain. So these are also excreted through the urine because of its poor absorption by the kidney. So this is the basic things of the phenylketonuria. This is the basic actually the disease which is actually happened in our body, the phenylketonuria. Okay, so this is the people with the PQ have a defective pH enzyme. So name of the enzyme I am repeating again, phenylalanine hydroxylase. I am repeating again, phenylalanine hydroxylase. When this phenylalanine hydroxylase lacks present in our body, so that time actually what is the main function of this enzyme? The main function of this enzyme that it's actually break down the amino acid phenylalanine into the tyrosine in liver. But when this enzyme is not there in our body, then what happen? Then phenylalanine become accumulated in our brain, and for this accumulation mental retardation takes place. Okay, next thing is the color blindness. Next disease is the color blindness. So this is the X-linked disorder. This is the X-linked disorder, one of the most important X-linked disorder also, like the hemophilia, where it results in the defect in either red or green cone of the eye. Okay, so in this disease the person cannot identify few colors. Okay, so it does not mean not seeing any color at all, but it actually in fact it leads to the failure in discrimination of color at all. Okay, between the red and green color. So the patients of the patient of the color blindness cannot discriminate, cannot separate the red and green color. Okay, because this gene of color blindness is present on the X chromosome. So that's why this is the X-linked disorder. That's why this is called the X-linked disorder. And the normal human retina contains the two kinds of light sensitive cell. I think all of you know that the rod cell and the cone cell. The rod cell is active only in the low light and the cone cell active in normal daylight. Okay, and there are two chemical pigment is also there. The rod cell actually synthesize the rhodopsin and the cone cell actually synthesize the synopsin. Okay, so these two cells actually responsible for the color perception. So normally there are three kinds of things, that the cones each one sensitive to a specific range of wavelengths. The red cone 64%, green cones 32% and the blue cones 2%. So it's present mostly in the males because of the presence of only one chromosome as compared to chromosomes in the females. Okay, and heterozygous female has normal vision but its carrier and passes on the disorder to some of her sons. So normally according to the pedigree chart, according to the pedigree analysis, the color blindness actually pass or transmit to the son rather than the daughter. So this is the basic thing. This is the types of color blindness. So first one is the monochromatism. Okay, so this condition actually complete the color blindness in which all colors appear as shades of gray. Okay, next is the dichromatism. So this condition of the partial color blindness in which only two colors are perceptible, red green and blue yellow. Okay, and the daltonism, this condition of color blind person who unable to identify red and green color. So this is the basic thing. And next thing is the protanopia. Okay, so this color blindness characterized by defective perception of the red and confusion of red with green or bluish green. Okay, and next is the deuteranopia, a form of the color blindness characterized by the insensitivity to green. So these things are not included in your syllabus. For your better understanding, for your knowledge, actually I have made this slide. So the main thing from your color blindness, this portion, that first you should know what is the main important thing, that color blindness is what type of disease, X-linked or autosomal. So first, this color blindness is the X-linked recessive disorder. And this what happened in the color blindness, this actually result in the defect of the either red or green cone of the eye. So who are a color blind patient, so they cannot actually discriminate between red and green color. Okay, so the gene of the color blindness actually present on the X chromosome. Okay, and normally this happen in the daughter. Okay, the son rather than the daughter, because this is the female, actually it's present mostly in the males because of the presence of the only one X chromosome as compared to the two chromosomes in females. So this is your today's class. In our next class we will discuss some chromosomal disorder, and we will complete this chapter with our last session. Okay, so in this session actually we got to know several things. The first thing is the how many types of disease is there. Okay, how many types of genetic disorders are there, and their types of genetic disorder, their causes of this disorder. Okay, several types of diseases there, and with their pedigree chart analysis and their normal genetic cause. Okay, so I hope this video will help you a lot for better understanding. But if you have any queries, ask me immediately. Okay, and in our next session we will discuss the chromosomal disorder. Okay, thank you, bye.